Explain the process of fertilization. Fertilization is a process where an egg cell and a sperm cell come together. The egg cell is much bigger than the sperm cell, and about 200 to 400 million sperm cells will be competing to make it to that one egg cell. Only a couple hundred of those sperm cells will actually make it. For the sperm cell to get to the egg cell, it has to swim, so it has a flagellum. Fun fact, the size of the sperm cell compared to the distance it has to swim would be like a human swimming approximately 40 miles. Now once that sperm cell makes it to the egg cell, it has to get through a couple layers. The egg cell has an outer jelly layer, within that a vitelline envelope, and within that the plasma membrane. The sperm cell has to get to that plasma membrane, and to do so it has an acrosome at the front of its head that contains enzymes that will allow it to break through those outer layers. The egg cell's plasma membrane has receptors that will identify the sperm cell. Once that first sperm cell makes it to the plasma membrane of the egg cell, the plasma membrane of the sperm cell and the plasma membrane of the egg cell will come together, and after that happens, there will be changes in the plasma membrane of the egg cell that will not allow any other sperm cells to make it through. Now inside the egg cell, the nucleus of the sperm cell and the nucleus of the egg cell will come together and share their genetic material in one cell. That genetic material will establish many of the characteristics that will be seen in that organism, like if the organism will be a male or a female. That one cell is called a zygote, and once that zygote forms, that ends the process of fertilization, and the process of cleavage will then take over.
Explain the process of cleavage.
Hi, my name is Chef Cleavage, and I'm going to explain to you my role in the embryonic development. Cleavage follows fertilization, and it's basically the rapid cell division of the cell into multiple daughter cells. When the cell divides, the cytoplasm divides with it as well. So let's imagine this grapefruit is a zygote, and it starts dividing in half, and then the two halves get divided into two other halves, into smaller daughter cells, and it keeps dividing. And so on. So once we get to 30 daughter cells or more, it's called a morolla. Now each little dot, these black dots on each grapefruit, are basically a regulatory molecules that house different genes, and they determine what the fate of each blastomere at the end.
The cell only divides, it does not grow, it stays at the same size, it just divides into smaller daughter cells.
So each little cell is called a blastomere, and each blastomere has these little regulatory molecules called cytoplasmic determinants, and basically each molecule houses different genes that determine the fate of each cell at the end of development. The cell keeps dividing until we basically get a clump of daughter cells together. They form a blastula. Each daughter cell is called a blastomere.
And of cleavage, we basically end up with a clump of daughter cells together. There are the blastula. And on the inside of the blastula, we have what we call a blastocele, which is basically a cavity filled with cytoplasm. And after this step, gastrulation begins, and where the 3 germ layers form.
Explain the process of gastrulation through detailed drawings. So now we talk about gastrulation. Gastrulation begins with the phase next to the blastula. As we see here, this layer is the trophoblast, which is responsible for nourishing the blastula, and which becomes after that placenta. Now we have the inner mass, cell mass here, which are going to be 2 layers, 1 and 2. Now I want you to take only these 2 layers, that's what we are about here, and see them in this picture. So we have 2 layers, that is bilaminar disc.
Explain the process of organogenesis. Organogenesis is the further specialization of the three germ layers: endoderm, ectoderm, and mesoderm. It's also the laying down of the body's major tissues and organs. An important component to organogenesis is the formation of the notochord, and the notochord gives rise to the body's nervous system, which consists of the brain and the spinal cord. Another important factor to the formation of the notochord is somites, which are a precursor to skin, bone, and muscle. So as you can see here, different concentrations of cytoplasmic determination decide which organs are actually formed in the body, and that is through a process called induction. And induction consists of an organizer cell, which tells the cells where to go, and the organizer cell releases morphogen, which is a chemical signal made of auxin and glycoid, which tells the cells what to become. Organogenesis is, in sum, the differentiation, proliferation, and movement of cells into a more determined state.
Oh, that's right.